Hello student, good morning. Today we will start the chromosomal aberrations. We have to study the last lecture, that is mutagens and chemical mutagens. Today we will start the chromosomal aberrations. The chromosomal aberrations as involves a change in the structure, that is structural aberrations, and numerical change in the chromosomes, that is numerical aberrations. These both are responsible for the chromosomal mutations, which affects the longer portion of chromosomes. Longer portion of chromosomes are observed in natural populations. Structural changes can be following types. Deficiency involves loss of a part of chromosomes. Deficiency involves loss of the part of chromosomes. Duplication involves addition of part of chromosomes. Translocation means involves exchange of segments between non-homologous chromosomes. Inversion, it involves the reserve order change in the genes of a part of chromosomes. Deficiency, number one is deficiency, that is the structural aberrations. Deficiency, duplication, translocation and inversion. Number one, deficiency. Deficiency is due to loss of a pad of a chromosome. Smaller deficiencies present in heterozygous conditions on one homologous chromosomes only can be tolerated by an organisms. Deficiencies are having an effect on the process of inheritance. Deficiencies on chromosomes in males and Drosophila were studied first by Stadler, LJ. In case of Drosophila, the deficiencies were recorded particularly on X chromosomes in the regions of genes capital W white-eyed, small fa facet eye, and capital V vermilion colored eye. The deficiencies are also recorded from mice and human beings also. Example, child with cat like cry, small head and low mental faculty. Low mental faculty. Chromosomes pairing in the deficiency heterozygote. A pair of homologous deficiency and patching. Second number, duplications. What is meant by duplications? These are obtained due to the addition of a part of chromosome. If duplication is present only on one of the two homologous chromosomes at meiosis patching state. Cytological observations, characteristics of deficiency will be obtained in duplication also. A pair of homologous chromosomes, duplication, heterozygote, patching configurations. Duplication are of different types. Tendon duplication is in the adjacent region where the duplication occurs. normal displaced duplication is on the different arm of the same chromosomes. Alternate orientation, alternate chromosomes will be oriented towards the same pole. In other words, adjacent chromosomes will orient towards the opposite poles. Second number, adjacent. First, in adjacent first orientation, adjacent chromosomes having non-homologous centromeres will orient towards the same pole. In other words, chromosomes having homologous centromeres will orient towards opposite poles. Adjacent second. An adjacent second orientation, adjacent chromosomes having homologous centromeres will orient towards the same pole. As shown in figure, alternate disjunction will give functional or fertile gametes. Adjacent first and adjacent second disjunctions will form gametes which would carry duplications or deficiencies, and as result would be non-functional or sterile. Therefore, in a plant with a translocation in heterozygous conditions will be considerable for pollen sterility. Translocation helps in the process of formation of new species. Inversion. An inversion is produced when there are two breaks in a chromosomes and the intercalary segment reunites in reverse order, that is, the segment rotates at 180 degree centigrade. 180 degree. Let's imagine that chromosomes will give rise to another chromosomal having order. The segments have rotated, there are 180 degree, giving an inverted order of gametes, genes. As similar hypothetical example using the chromosomes ABCDEF has been shown in figure, where due to coiling breaks occurs between B and C as well between D and E. Reunion at broken ends may lead into inversion of segment CD into DC. ABCDEF, ABEF DC, ABEF DC, ABDCEF. Only of the possible mechanism which may give rise to the chromosomal inversion. Inversions can be two types. Inversion.